Okay, so good afternoon, everybody, and we're going to carry on our discussion today about viruses. We'll finish our discussion about viral biology and then talk about a couple of viral diseases. I just want to remind you, please, today is the only class that you're going to have for this week because of Thanksgiving. But I want to remind you, please, that a number of you have not yet submitted your written questions from your test. Please do so by 4 o'clock today. Any that come in after 4 o'clock today are going to lose points for being submitted late. Those that have been sent to me attached to an email as a PDF or a Word document, not as an image taken by your phone. Okay. All right, so let's carry on with our discussion about viral biology. This is where we finished off last time with discussion about the bacteriophages, viruses that infect bacteria, that are able to enter into one of two types of life cycle. And the first kind of viruses that we saw were the lytic viruses, which simply infected a cell and then ended up bursting the cell to release all of the new viral particles. This virus here, bacteriophage Lambda, is a bit different, and it has a characteristic which is common to many of the bacteriophages, but also bears some resemblance to some of the behavior of some animal viruses in a way. And we refer to this as the lysogenic cycle. That is, the virus is able to produce a lytic cycle, just as we saw before. It infects, causes the bacterial cell to become a viral factory, ends up bursting, lysing, and releasing viral particles. But the lysogenic viruses are also able to go into another silent life cycle. And this is the lysogenic cycle. In the lysogenic cycle, the viral DNA is integrated with the host DNA, and it forms a section called the prophage. All it is DNA. That's all it is. It's just got all of the genetic information to form viral particles, but it remains silent. And every time the bacterial host cell replicates, it replicates the viral genome as well. Until a certain point is reached, it's usually an environmental cue of one sort or another. And at that point, when the correct signals are received, the viral genome loops out and begins to act in a lytic fashion, just as we saw before. So what we have is an alternation. There's an infection, a virus remains silent, lodged as a prophage. Then at some point or another, it goes into the lytic phase and ends up reproducing and being released. Now, I say that has some resemblance to some animal viruses because there are viruses in animals which are able to remain in a latent phase. And they very often do so by doing exactly this, by integrating with the host DNA and not actually being reproduced by the cell until such time as some environmental cue is reached. It's a similar kind of system. Okay, so let's have talk a little bit about animal viruses. And these, of course, are of particular interest to us because these are how our viruses behave. And there are some similarities to those bacteriophages that we saw, because first of all, the very first step of any viral infection is that the virus has to bind to the correct host cell and recognizes the correct host cell by binding to specific receptors in the host membrane. And though that attachment is effected by the outside of the virus, we saw that there were some viruses which are naked, just a protein capsid, or there are also viruses which are enveloped. It doesn't matter. In both of them, there are viral components, viral proteins, which are responsible for recognizing specific receptors on the host membrane. That's what gives viruses their specificity. If they are unable to bind, they are unable to proceed with infection. And so once they have bound and been able to enter, then there is a period when the virus genome has to be liberated from all of the rest of the viral particle. So, for example, if it is surrounded by a capsid, the capsid is divested from the genome. At the same time, remember that what is contained in the capsid may not only be the genome. Sometimes, it depends on the virus, sometimes there are essential viral enzymes and proteins which are included, which are also liberated into the cytoplasm of the host once that uncoating is performed. That uncoating, by the way, there's different mechanisms to it. Sometimes the uncoating, the dissolution of the capsid and everything is affected by host enzymes, but there are some viruses which have their own enzymes which are responsible for the uncoating. The next thing is that it depends very much on the virus as to what happens now, but in all viruses, what they're going to do is they're going to co-opt the metabolic and genetic machinery of the host cell. And they're going to get the host cell to turn into a little viral factory. They're going to begin biosynthesis of all of the viral components, the viral proteins, the viral genome, any of the viral enzymes that must be included in the capsid, et cetera, et cetera, et cetera. Then once all of those are complete, a complex period of maturation begins, when all of these components must be assembled, and they are assembled into the correct order. The capsids are created, for example. Essentially, the capsids kind of crystallize out. The genome has to be properly packed. And then once the particles are ready, they need somehow to be evacuated from the host cell. They need to, and in animal cells, there are two principal ways in which this occurs. The first is that the virus particles can actually bud out. We'll see this in the diagram in a minute. Or else, it's the old familiar thing that we've heard about already. The host cell is induced to actually rupture, to lyse, and release the viral particles. It depends on the virus, on its particular life cycle. Okay, so let's have a look, first of all, at some of the entrance mechanisms, because they are of particular interest to us at the moment. There are two ways, principally, in which viruses enter animal cells. The first is actually by being swallowed up by the cell. What they do is they actually, in the viral proteins, induce the host membrane to form an endocytic vesicle, just as if the cell was swallowing it from the outside. So that is a process of endocytosis. This is of great interest to us because this is exactly how COVID-19 enters our cells, is by this mechanism. Once that vesicle enters like this, then the vesicle is removed in one way or another, and then the uncoating of the virus particle begins. Now, there's another mechanism, and that is that in many animal viruses rather, are surrounded by an envelope, as we saw before. And very frequently, one of the functions of that envelope is to bind to host membrane. So here is the cell membrane of the host. Here is the membrane around the capsid in the center here. This is, in fact, I think that this is HIV that they're trying to show here. Here's the capsid here with all the innards the genome and everything inside here. This on the outside is an envelope, which is derived originally from some kind of host membrane. And it's very easy for this membrane to bind to the host, to the membrane. And once it has, it opens up, it unites with it completely, and it actually injects the capsid into the cytoplasm. So this is the non-endocytic route. It's fusion of the virus at the cell membrane. So here they are, both of them. These are neat electron micrographs. Here's the host membrane. This is a virus which is inducing endocytosis. So endocytosis is actually induced by the layering of specific proteins called the clathrins. And the clathrins, once they lay down, they actually automatically form a sphere. And in doing so, they pull the virus in a vesicle. This here is herpes virus, and here it is. It's surrounded by. It's not that easy to see here, but it's actually surrounded by a membrane, and the membrane is united. It's uniting there. This is the capsid here. This deeper orange here. Here's the membrane around the outside, and it's uniting here to inject the capsid. Now, similarly, I mentioned there are different ways for the viral particle to exit, and this is the way in which, for example, a viral capsid will bud out from host membrane. Now, it may be that, as is shown in this diagram, that this is the host cell membrane here, and as the viral particle exits, it forms a bud, and it leaves surrounded by an envelope. So this is the mechanism for many, many animal viruses to leave the host cell. But I will just mention that this does two things. First of all, it gets the viral capsid outside, of course, to go and do whatever. But second of all, it invests the viral capsid with the envelope. And this is the origin of the enveloped viruses. This is where they derive there. But this diagram here only shows this viral particle leaving through the host membrane. Well, that is very often the case. But the membrane of the enveloped viruses may be derived from other sources inside the host cell. So, for example, we're going to see in the COVID-19 rather, the SARS coronaviruses are actually derive their membranes from endoplasmic reticulum, not from the cell membrane. They still leave by budding, but their initial envelope is derived from endoplasmic reticulum. So the envelope that we see around the enveloped viruses can have various sources. It depends on the virus. It depends on the particular virus we're talking about. We will also talk in a little while, a little bit more detail about the fact the viruses don't only cause sorts of acute diseases that we're used to, influenza, corona, COVID, et cetera, Viruses also often implicated in tumor-forming processes. They are strongly associated with a number of different cancers. And for example, we know that sarcomas and adenocarcinomas, these are both cancers which are associated with viruses. And one of the mechanisms that viruses actually change cells so that they become tumor forming is by interacting with specific genes in perfectly normal cells. Now, our genome is full of regulatory genes. For example, there are all sorts of genes which regulate growth. There are all sorts of genes which are involved with cell division. They are measuring stuff that is happening inside of the cell, stuff from the outside. They're measuring the environmental conditions, all sorts of things. And in response to those, they are regulating the correct growth and the correct speed and timing of cell divisions, etc. Those all have the potential for going wrong. And if they went wrong, then the cell loses control over the growth mechanisms. Those genes which have that potential, we refer to as the oncogenes. They have the potential to be transformed, to be changed, so that normal cell function is lost and the cell is transformed. That is the proper term for it. It is if we say a cell has been transformed, we imply that it has been changed from a normal cell into a cancerous cell. And the oncogenic viruses are viruses which become integrated into the host's DNA. And if they are integrated near to or in an oncogene, they actually change the oncogene function. And the cell loses control over the function. I'll give you one example. It is very frequent in cancer cells that growth regulation is disrupted. And one of the things that you see in cancer cells is that you get many different sizes, many different kinds of cell developing, all because oncogenic virus has disrupted the genes responsible for growth regulation. So one of the ones that we will hear about is Papilloma Virus. And the Papilloma Virus, there are many strains of Papilloma Virus, but in general, they cause a completely harmless, benign tumor, which we recognize as a wart. But under certain circumstances, they are strongly oncogenic. They change cells, transform them into cancer cells. And this is particularly the case in cervical cancer, also in penile cancer. But penile cancer is very much less common than cervical cancer. So there are a long list of these oncogenic viruses, many different kinds. And in fact, more and more of these kind of oncogenic functions are being identified. And this is an area of really major research. Okay, there's a couple of other things to mention about mechanisms of viral disease. And the first is we need to draw a distinction between viruses which form an acute infection and are then eliminated completely and disappear. And there are viruses in animal viruses which are able to enter into long periods of latency. And the most important group in human beings are the herpes viruses. This is a characteristic of the herpes viruses. They are able to integrate into the host genome, into the DNA, and remain there often completely silent for the host's entire life. But under special circumstances and very often very poorly understood circumstances, they become active again and cause a disease. The simplest example of the herpes viruses that cause cold sores around your mouth. And we'll see this again in a while. But other herpes viruses like chickenpox, for example, can re-erupt many, many years later to form a separate disease called shingles. So, and that is only one example of these kinds of viruses. Now, the latency has to be distinguished from another type of viral activity, which is persistent viral infection. Now, this is again of some interest at the moment because there is some indication that COVID virus, Sars Kovi Ii, which is the virus that causes COVID-19, there is some indication that this may be part of its behavior, possible behavior. But persistent viral infection is not a latent infection. It's just a very, very low grade, continual reproduction of the virus. And there is one which is particularly dangerous. Fortunately, it's rather rare. And this is called subacute sclerosing panencephalitis. And this is a disease, a neurodegenerative disease, which results from measles infection. And it is one of the reasons why measles vaccination is so important, because this subacute sclerosing panencephalitis can make itself apparent many years after an initial childhood measles infection. When the person is a teenager or in their 20s, they enter into a period of neurodegenerative disease as a result. And there's very little that can be done. Fortunately, it is rare, and has become vanishingly rare with vaccination. It is where a vaccination against measles is widespread. This is very, very seldom seen anymore. Here are a range of kind of latent and persistent viral infections. The cold sores, obviously, you understand. Cold sores are a latent infection because the genome, the viral genome, actually integrates into the DNA of nerve cells. And when it's activated, it makes its appearance at nerve endings around the mouth. It's very, very similar to the viruses that cause genital herpes as well. And in fact, you can get either. The genital herpes is a strain of the cold sore virus. Shingles, again, I mentioned, is a result of chickenpox infection, and this is a latent infection, again, of nerve endings, and tends to make its second appearance either on the face or around the trunk of the body. Cervical cancer, this is a papilloma virus, and the papilloma virus, again, is able to act on the oncogenic nature of cells and transform them. HIV/AIDS, there is some contention as to whether it is a latent infection or whether it is a persistent infection. But if essentially it has a long period where it appears that it is more or less latent, in that it becomes integrated to cells of the immune system especially, and only activates when those cells are activated. That's its main route of disease, of pathogenesis, is it integrates into T cells, especially in the immune system. And once they are activated in one way or another, the virus is activated along with them, et cetera, et cetera. There are several of these just for interest's sake. The last thing I'm going to tell you about, forget about the viruses just for a minute. We're going to return to viruses very shortly to talk about viral disease. But the last thing I will mention is not a virus at all. It's got nothing to do with viruses. But this is a very, very strange thing. And this is actually just infectious protein particles. And these are called prions, proteinaceous infectious particles. So it's prion, and the prions are very strange and rather disturbing because they're just a complex chemical. That is all. They're a complex protein, and they have the ability to transform proteins which strongly resemble them. So what we have is a set of proteins which are essential, especially in the nerve cells and things, let's say. We have these proteins which are essential, normally produced by the metabolism, et cetera, and are used, but there are mutant forms of them. And the mutant forms, once they enter the cell, are able to convert the normal proteins into the mutant form. They induce a structural change in other proteins in the cell, and they end up disabling the cell entirely. And again, these are not common diseases, fortunately. They are more common in animals than they are in humans, but they unfortunately are transmissible from animals to humans. And they cause a set of diseases which are commonly referred to as the spongy form encephalopathies. And this is because literally the brain, especially when you examine it on post-mortem, it has a spongy texture because cells have been destroyed as these proteins have multiplied and destroyed cell function. So the most famous one, of course, is mad cow disease. And a mad cow disease is a disease of cows, but it is transmissible to human beings. One of the characteristics of these prions is that they resist heating. Heating them, autoclaving them, and cooking them makes no difference to them. And if you continually ingest these proteins, there is a possibility that you end up, some of them may eventually end up in your nerve tissues. And once they are, you're infected with them, they proceed to cause this disease. And in humans, they cause this disease here. It's called Kreutzfeld-Jakob disease. And it is a paralytic neuropathological disease. There are various other ones, which formerly their causes were unknown, but now we are beginning to look at them and realize that they are from these very strange protein particles. Okay, so just give me 1 second, please. That is all that I need you to know about prions for the time being. And hold on 1 second. What happened? I just hope. Let me just see. Oh dear. What have I done? All right, hold on 1 second, really. I just hope that you can see this. I'm going to talk to you a little bit about some of the viral infections. This is just really no more than an introduction to viral infections, just so that you hear about some of the most important. But in doing so, we'll hear some of the principles of viral involvement in human infections. But realize that we could have a 2-semester series of lectures on viral disease. It's a very, very complex topic indeed. This is nothing more than the briefest of surveys to spark your interest. Okay, so I want to first of all start talking about these upper respiratory tract and lower respiratory tract infections that absorb so much of our attention. They do anyway, but of course nowadays with the great pandemic raging, our attention is focused on viruses which cause respiratory disease. Well, the very first thing I want to tell you about is the common cold, because believe it or not, the common cold, for all that we are used to it, we're used to getting the common cold. There is tremendous amount of confusion about it, and despite the fact that we think of it as just being a relatively mild thing, it causes a tremendous amount of economic loss every year, and can in fact, especially if it's not properly treated and not respected, then it ends up producing a tremendous loss of work time and everything else, and also exposes us to the risk of secondary infection. Even the common cold, just its very name suggests that we shouldn't take it that seriously. Well, we should, because it does have, first of all, it can make you feel truly miserable. Don't belittle somebody and says they've got a cold and say, well, if you've just got a cold, you should be able to come to work. No, it's a viral infection. It is spread very, very, very easily. and could cause a tremendous amount of lost time as the disease spreads through a population. Now, there are confusions with the common cold and things like influenza. And the reason for that is they share some symptoms, but they don't share all of their symptoms. The first thing is that the common cold, usually it has quite a long prodromal phase. Look back in our epidemiology and disease process lecture. And the prodromal phase is when the person who has been infected realizes that there is something going on. They have the very mildest of symptoms, which slowly increase, but they are aware they're not quite right, but they're not sure what's going on. Common cold usually has a lengthy prodromal phase, a prodromal phase of days, usually. That distinguishes it from other diseases like influenza, which has a very short prodromal phase. Very often is accompanied by a sore throat. It's the commonest symptoms of the common cold are all upper respiratory. There you have a runny nose, often runny eyes. It affects the mucous membranes of the head primarily. It is not usually accompanied by major fever. There may be a mild fever, but it's very, very seldom. Very often with common cold, you have no fever at all. Seldom have anything more than the mildest of headaches. And there may be some muscle ache and fatigue. But really, it's the runny nose and then sometimes a cough. But the cough in the common cold is seldom produced by the virus itself. It's a severe cough associated with the common cold is usually an indication that there may be a secondary bacterial infection present. So that's the common cold. The common cold is caused, by the way, by anything up to 200 different viruses that will produce those same symptoms. So there's a huge range of different viruses that will do exactly the same thing and produce symptoms which are virtually indistinguishable. The commonest is this virus here. This is called the Rhinovirus, and rhino means nose. So it's no surprise that it's called the Rhinovirus. And it's the commonest cause of the common cold. But as I say, there are many, many others that can do the same thing. They have an interesting ecology because the rhinoviruses and other common cold viruses will actually infect the nose and sinuses and upper respiratory tract because the temperature is lower there, slightly lower, about 33 degrees Celsius. They don't thrive at a higher temperature. And they are very, very easily transmitted. These are viruses which survive quite well on fomites, for example. So people sneeze unguardedly or wipe their noses and then put their hands on a surface. The rhinoviruses, for example, will survive quite well for quite a long time. One of the major transmissions of the common cold during cold season is through the hands. And it can very definitely be transmitted through droplets when people sneeze and cough into the air. That is true. But one of the commonest means of transmission is on the hands, and then people scratch their eyes, pick their noses, whatever. Any introduction of the virus to mucous membranes provides a route of infection. So these are things to be very careful of during the common cold season. During common colds, there is very definitely a common cold season in the cooler months of the year. Exactly why is not known. It has been suggested that one of the major reasons is that people tend to spend more time indoors and close to one another during the cold parts of the year. It may be that the virus survives in droplets for longer in the cooler weather, or something like that. But spending more time indoors and more time crowded with other people seems to be one of the principal reasons that the cold is more common during the winter months. But it certainly occurs throughout the year. And you've all probably had that experience. We can pick up a cold any time of the year. They are constantly around, constantly being shuttled between people, and then blooming usually in the autumn and the winter months. There are another group of viruses which also are important in the common cold, and these are the adenoviruses. Now, the adenoviruses sometimes operate a bit differently, and they can, in fact, cause a severe sore throat and some fever as well. So these, if you have a common cold and you have a higher fever with a cough, and it's sure you've had it examined and you've had a culture done, whatever, and it's sure that it's not bacterial, it is very likely one of these adenoviruses that is causing your illness. The reason we pay particular attention to adenoviruses is in young people, they're seldom of any problem and honestly quite rare. But in elderly people or in immune-compromised people, the adenoviruses can be extremely severe and can progress to a pneumonia unassociated with bacteria. It's a viral pneumonia caused by the virus itself. Very, very uncommon in people with a normal or healthy immune system. Okay, so we need to distinguish those upper respiratory tract infections from here on up from those of the lower respiratory tract. And the diseases of the lower respiratory tract tend to be far more serious. Our lower respiratory tract is kept largely sterile, and that is through the continual action of the bronchomucociliary escalator, which moves a sheet of the mucus continuously through the lower respiratory tract up to the top of the throat where it is swallowed. That sweeps out particles. It sweeps out debris, it sweeps out bacteria, and it also sweeps out viruses, hopefully. But there are those which can infect the lower respiratory tract. And the influenza virus is one of them. Now, the influenza virus and the common cold virus are very commonly confused. And people, for example, with a common cold, even though we call it the common cold and we're used to having it around and everything, it can make us feel extremely ill. And when we do suffer from severe illness from the common cold, where very often people will say, I've had the most terrible flu, I've had terrible influenza, where it has in fact got absolutely nothing to do with influenza. Influenza viruses are completely different from the common cold, all of the common cold viruses. And the influenza viruses are a very complex group of viruses with distinct structural differences that we can distinguish them from all other viruses. Second of all, the progress of an influenza infection is somewhat different. First of all, in influenza, the person tends to have a very, very short prodromal phase of the illness. A matter of a half an hour or an hour can make the difference between essentially feeling quite well and feeling extremely ill. And the onset of influenza is very frequently marked by the development of severe headache, which seldom occurs in the common cold. The second thing is that influenza tends to produce fever. And it also produces much more profound muscle ache and fatigue. Fatigue, headache, and temperature are the kind of hallmarks of an influenza infection. which distinguish it from common cold. So influenza is a very communicable disease. It's transmitted, as you know, by airborne respiratory droplets. And it has the characteristic that we know that it periodically moves through the human population in great pandemics that sweep through the human population, many of them originating in the Far East. We know that there have been at least 31 influenza epidemics since 1510, which is the symptoms of influenza were recognized even earlier than that. They're in some of the best medical writing is of Arab doctors. And I believe that there are records as far back as the 800s detailing symptoms which might have been influenza. We don't know exactly when it entered the human population. Here's the interesting thing about influenza, about its biology, is that influenza principally has three separate host groups. And those are pigs and humans and poultry. And each of those groups, they have their own influenza viruses that are endemic to those groups and circulate easily through those groups. However, there can be crossover. And the crossover is especially important between birds and humans, with pigs acting as intermediary. And pigs end up being able to acquire either both viruses, human and birds, or acquiring a hybrid virus and then mixing it and producing a virus which is easily transmitted to humans and then from human to human. We can also pick up the virus directly from birds. So we have this kind of complex movement between these three reservoirs. And when it moves through those three reservoirs, very, very bad things can happen. Because when it emerges from the reservoir and enters the new host population, it can cause a devastating pandemic because there is no resistance to it. So it's primarily a zoonotic disease. Influenza is a zoonotic disease. We acquire it essentially from animal reservoirs, from poultry and from pigs. But we also have our own influenza viruses. So I've done most of this, I think. The danger with influenza is that it does not remain as an upper respiratory tract infection. It can move to the lower respiratory, it can cause lower respiratory tract symptoms as well. And it is really a systemic illness. It is not isolated to the respiratory tract. In fact, you can have all sorts of symptoms of gastrointestinal distress and all sorts of things associated with the infection as well. It can be an extremely dangerous infection. And the reason for that is that in human beings, it can excite a very, very strong immune response. And it is in fact the immune response very often, which results in high morbidity and mortality from influenza. So it's an enveloped virus. Here it is here. And it's an RNA virus. Here's the envelope here around the outside. And here's the capsid here. These little 8 things here. There. Each of these is enclosed in a capsid. So it's a very strange organization. There's this, there is a protein shell which is not really capsid, which lies underneath the membrane there. And inside, the genome is divided into 8 segments. each with its own capsid around it. Very, very strange organization. What is really, one of the things that's really important is that embedded from this protein layer here, and then through the membrane, are these proteins, these spikes sticking out. And we recognize two kinds. One is called Neuraminidase, and the other one is called hemagglutinin. And hemagglutinin is the principal attachment protein. So we recognize there are different kinds of hemagglutinin. There's currently, I say currently because we keep on expanding our knowledge of them. There are currently 17 kinds of hemagglutinin. The neuraminidase has actually a function to on release of the viral particle, and they are responsible for getting the viral particles out of cells once they've been fully assembled inside the host cell. So the one hemagglutinin for entry, neuraminidase for exit. There are 10 kinds of neuraminidase. So when we look at a flu virus, we look at the type of H and the type of N, and we actually number them. We can number them according to the H and N proteins that they have. In addition, I should tell you that there are 3 broad groups of these viruses, but there's only one group that we are particularly interested in. The first group is Influenza A, and that we get virtually every year. This is what is responsible for the most vast number of epidemics and pandemics that we see every year. Influenza B is a lot. Also, you do see it each year, but it's rather uncommon compared to type A. And the last one is Influenza C. It's a very mild disease, and it tends to drift through the population throughout the year, and it's not really recognized as being an epidemic illness. There are, apart from the fact that the influenza itself is a very serious illness, it really has a high morbidity, and quite apart from that, it can set up a situation where you are very prone to secondary infection. But there are a couple of other rare outcomes of influenza. The first is Guillain-Barre Syndrome, which is a paralytic syndrome, which is probably autoimmune and resulting from our immune system actually attacking our own nerve cells after a viral infection, or sometimes very rarely after a vaccine. Reye's syndrome is one the parents among you should pay particular attention to. Be very, very wary of using aspirin when your children are ill, because if they have influenza, they may develop this syndrome called Reye's syndrome, which can be extremely serious. So acetaminophen is a preferred anti-inflammatory if you wish to do so. All right, so here's the thing. Every year we get told to go and get our flu vaccines. And the question then is, well, why do we have to get a flu vaccine every year? Why can't we just have a single flu vaccination and have that cover us for the rest of our lives the way that other vaccines seem to work? The truth of the matter is that the flu vaccine is actually a very good vaccine. In the years where it works extremely well. Influenza is a very dangerous disease. It carries a really quite high morbidity. It has become so much a part of the human population that we kind of disregard it, but we discard it at our peril, because influenza causes a number of deaths, and not only amongst the elderly and the immune compromised, it also causes deaths in young, healthy people. Not commonly, but it does, and it very often does so through the overexcitation of the immune system, resulting in a shock situation where there's a catastrophic loss of blood volume, of blood pressure rather, and sometimes also a disruption of blood clotting systems. So it has the potential to be dangerous. We really should have our flu vaccines regularly, not only to protect ourselves, but to limit the epidemic nature of flu each year. But why each year? The answer is that this is a very, very mutagenic virus. This is a virus which changes very, very easily. It mutates extremely easily. And the principal area of mutation that happens each year is in those H and N spikes around the outside. And this is what our vaccines are designed to do. They are designed to recognize those H and N spikes. When they are able to, when the vaccines are able to produce antibody which recognizes those spikes, the vaccine works extremely well. But each year, as this virus moves through the human population, either in epidemic or in pandemic nature, those mutations slowly accumulate, and those H and N spikes change ever so slightly. And even a small change in the H and N spike renders it invisible to the immune system. So each year, a committee of scientists gets together and they examine influenza strains from all over the world. You see little minor epidemics develop in Sydney, Australia, or in Hong Kong and in Paris and wherever. And they look at these and they look at the epidemiology of it, the spread and everything else, how infectious it appears to be. And they choose usually 3 strains of influenza that are then put into vaccine production. So when you go and get your influenza vaccine each year, hopefully the committee have chosen the 3 strains that you are most likely to be exposed to. Sometimes, obviously, they get it wrong. But most years, we gain at least some protection. And some years we get very, very good protection. Last year, I think, the protection was very, very good. So that slow change of the H and N spikes is called antigenic drift. Antigen is something that your antibody sees. So the H and N spikes, those are antigens, which your immune system is going to recognize and produce antibody against. The antigenic drift is the slow change of the antigen. Antigenic shift, however, is another process. It is abrupt. It causes major changes in the virus, and it essentially produces a new species of virus very, very quickly that then enters the human population, which has no resistance to it. This is primarily, by the way, of Influenza A viruses. We seldom see major problems with B and C. So here's essentially is the way that this works. In each of these, I told you, we have populations in poultry, especially ducks, but also in chickens as well. We have influenzas which primarily infect birds. We have influenzas which can infect pigs, but the important thing about pigs is they can acquire a virus from both human and from poultry. Humans have their own influenza viruses as well, but especially the Influenza A viruses tend to move between these 3 different reservoirs. And those 8 segments of the genome, each with their own little capsid, they can be mixed and matched in the process of moving between these reservoirs. So, for example, this will just explain it in simple form. Here's antigenic drift. Antigenic drift is simply the slow accumulation of small mutations, especially in the spikes, in the H and N spikes. As that happens, we need to have new vaccines to counter this, but these are essentially different strains of flu virus that have been seen before. However, when we look at our three reservoirs, at birds, at humans, and at pigs, what we very frequently see is, first of all, humans can acquire virus directly from birds. So humans can be infected with an avian virus. In addition, pigs can be infected with both human and with avian. And this is a particular danger because when two infections occur simultaneously, there is a chance that those are going to get mixed and that we're going to end up with a cross between avian and human mixed in the pig and then transmitted as a completely new virus to the human population. So we can acquire directly from birds, but very much more frequently what happens is we acquire recombined viruses derived from the different sources which have been mixed in pigs and then transmitted to humans. So if we look through human history, we can see when these new viruses enter the human populations. And there have been numerous of these pandemics, some of them absolutely disastrous to human beings. So one of the most important of all time was this epidemic here, which was called Spanish flu. It was nothing to do with Spain at all. It had nothing to do with Spain. This occurred during the First World War when there was strong censorship of newspapers, magazines, and everything else. And all over the world, they suppressed information about the influenza epidemic that was raging because they didn't want to discourage people in the middle of the war or whatever. But the only place where they allowed publication of information was in Spain. And so they were the people who were telling everybody there's this terrible epidemic. So it unfortunately got named the Spanish influenza. It was not Spanish at all. In fact, there is strong evidence that it began here in the United States and was then exported as soldiers moved all over the world to fight in the First World War and then returned with new mutants from overseas. So this is the Spanish flu, and just have a look at its origin. It was actually we believe, a direct transmission of a mutated avian influenza to humans. And it was an H1N1 influenza. And H1N1 influenzas still occur with regularity. And some of them can be traced back to this now not nearly as virulent, but there are definitely viruses which can be traced back to this original entry into the human population. So this was a direct bird to human transmission, and absolutely disastrous. And the entire, where it may have been a new recombination of avian and human, but all of these segments are believed to have been derived originally from bird. An absolutely disastrous pandemic. It caused mortalities which rival our current COVID pandemic, but remember that the human population was lots smaller then. So the impact was absolutely enormous. And it continued to make its reappearance until the 1920s. But it mostly had died out by the 1920s. The next major one here was called the Asian flu, and it was 1957, 1958. And this one is again an avian virus and a human virus, but this time it was a combination of the two. And the reassortment, when you look at the genome, you can find human segments of the genome and avian segments, And this took place in pigs, this reassortment. They got infected with both and ended up producing a new viral particle. Once it entered the human population, nobody had resistance to it, and it caused a great pandemic. In between, you can see there are regular entries of new viruses. This was the famous Hong Kong flu. Hong Kong flu had a very high morbidity, caused people to be extremely ill, and it did have a high mortality, as well. This again, this is an avian virus again, and here's a human virus, but now, interestingly enough, this human virus here may have been a holdover from this one here, this H2N2 that originally caused this Asian flu. So this is this virus getting remixed with another avian virus, again resorted in pigs and transmitted to the human population. And then this has been one of the latest ones. We did see a combination of all three, of swine, avian, and human virus. This ended up being a mixture of all three. It's the most recent. This, by the way, the novel H1N1. It's kind of a theoretical name that is given, but this is the great dread that this kind of reassortment is eventually going to produce a really virulent influenza virus. The ones that have appeared so far have not been that severe. There was one, I forget when, 2011 or something like that, which caused a great scare, but ended up not being particularly virulent, but was this kind of reassortment. But we can't tell. This is a great danger. We can't predict what impact they are going to have. Okay, so that brings us to, we need to discuss, of course, our current coronavirus pandemic. Now, this is caused by a virus in a group called the coronaviruses. These are positive strand mRNA viruses. Go back to our previous lecture and look up exactly what that means. They are positive strand mRNA enveloped viruses. And they're caused by this particular virus, which has been designated Sars Kovi Ii. And that is to distinguish it from a very, very similar virus called Sars Kovi I, that we'll talk about in a little bit more detail in a minute. This is what it looks like, probably absolutely familiar to you by now. This, you can see the envelope, the white envelope, that is the membrane derived from the host cell. We'll see how that happens in a minute. But embedded into that membrane are these spikes. And those spikes are important to us because that is what our vaccines are based on. Whatever vaccine we're talking about, we are trying to train our immune system to recognize these spikes, produce antibody, and inactivate the virus by just gumming it up with antibody before it can infect anything. So these coronaviruses tend to have the same kind of structure, and they get the name. Corona means crown. And when we look at them in the electron microscope, then this typical structure, the membrane with all these spikes sticking out, looks like a crown. That's how they got their name. There are a lot of coronaviruses that have been identified. And there are 4, for example, which are very well known causes of the common cold. Now that is very interesting because they are very mild. They just cause a mild common cold, and they move through the human population regularly. We've probably all had them at one time or another and just thought we had the sniffles. But maybe it is very, very possible that their original entry into the human population was far more severe, and that we have just adapted to them and they have adapted to us over evolutionary time until they're just these 4 mild coronaviruses. But then there's a whole set of coronaviruses which have entered the human population a lot more recently. And those are the viruses which cause catastrophic illness in human beings. And these are all zoonotic viruses, and they appear mostly to be derived from bats for some reason. Exactly what the association with bats is, we don't know, but we do know that bats have many, many, many coronaviruses that they are completely adapted to. They just maintain the populations as a persistent infection, or that if they do have an acute infection, they don't appear to be badly affected by it. So the viruses are maintained in bat populations constantly. The trouble with all of the coronaviruses is they tend to be promiscuous. That is, they can infect many different hosts, especially other mammals. And they can even temporarily move into a population and then die out in that population, only to reappear in another population of animals. So we see this with our coronavirus, for example. They can infect cats of many different kinds, for example. They can infect apes. All over the world, as zoo animals have been recorded as having acquired COVID-19. Usually fairly mildly, it's not catastrophic. But it's just an example of the fact that these coronaviruses can, in fact, move into a variety of hosts. Here are some of the really important ones. These are relatively recent entries into the human population, and some of their entry has been quite spectacular. This is Sars Kovi Ii, and this is the disease of SARS. Its animal host was never, the animal host which transmitted it to humans has never been definitively identified. But it was thought to have been another mammal. It's like a civet cat or something like that. There are other SARS viruses, coronavirus, which have been identified in pigs, for example, but we have no evidence of transmission to human beings. The other this, by the way, is a serious disease. We'll talk about it in a minute. This disease here is called Middle East Respiratory Syndrome, MERS. And MERS is a very serious illness with a very, very high mortality rate in human beings, and it is acquired from camels. Fortunately, its outbreaks have been extremely limited, and it causes such virulent illness that the epidemic itself is limited in its ability to spread. That is Middle East Respiratory Syndrome. Then we have our Sars Kovi Ii. And Sars Kovi Ii is very strongly related to this, but less virulent than Sars Kovi I. And it appears less contagious as well, fortunately. The others, there are many identified in other animals, but they don't appear to spread to humans. These are the particular ones. Apart from our 4 cold viruses, these are the ones that are of particular concern. So I'll just tell you a little bit about Sars Kovi I, because it was a disease that caused a tremendous amount of concern when it first erupted, because it seemed to have such potential for pandemic capability. And again, it was recognized as an emerging disease, very quickly identified as being a coronavirus. It was very, very virulent, and it had a high mortality, 14 to 15%. COVID is between 1 and 2%. And very, very high in people over 60, so 55% mortality in people over 60. So it was a very, very serious illness. But it very quickly, on infection, is so virulent, it quickly produces symptoms, which are easily recognized as being serious. So those people who acquired the disease tended to become very ill very quickly and were quickly put into hospital and put into isolation. And it is one of the features of the disease which limited its spread, allowed it allowed the epidemic to be controlled very quickly. It's can be spread through aerosol spray, but it can also be spread through fomite infection. The epidemic occurred in 2002, 2003. The origin is not exactly known. It is known that it arose in China, and it seemed to arise from a food market again, as is suspected in coronavirus too. Altogether, 8,000 people were infected very, very quickly. In 114 days, 774 of those people died. And the great tragedy was that many of the people who died were healthcare workers who were dealing with the patients without knowing what they were dealing with. Before it became apparent how very virulent and how infectious Sars Kovi Ii was, many healthcare workers had been exposed. And then once that was realized, then they instituted very, very rigorous isolation of people who had been exposed. There are differences to Sars Kovi II, which are fortunate. And first of all, in Sars Kovi II, our current COVID, there is a fairly long prodromal phase, and there are some marker symptoms like loss of smell, loss of taste, etc., which are so classic that people tend to isolate themselves or be isolated very, very quickly. In Sars Kovi I, people could be infectious. It was so infectious that even their short prodromal phase was enough to expose people to infectious dose very, very quickly. So here it arose in China, but the main center of spread was from Hong Kong, because it made its entry to Hong Kong from China. And then from Hong Kong, Sars Kovi I was spread very rapidly by air travel especially. There was some export to the United States, to Canada, and to Europe. But those places, it was already well recognized. The disease had already been well characterized. So when these cases arose, they were very swiftly isolated. In fact, they instituted quarantine for whole entire plane loads of people and everything. And they managed to contain it. All of that containment occurred because of the spectacular nature of the disease itself. This is not the case with COVID-2. This is not the case with our current COVID crisis. Our current COVID crisis, people can go through a relatively long prodromal phase. They can be infectious for a long time, and they can spread the disease without even knowing it. And in addition, of course, we know that some people never go to a doctor or whatever when they have COVID. They are able to treat it much as if it was a mild illness. But the danger with that is that they are in fact infectious and they spread the virus widely. So the disease has spread as a pandemic far more efficiently than Sars Kovi I ever did. But overall, even though it has a much lower mortality rate, because of the huge numbers of people that have been infected, the mortality is quite high, is really high. Millions of people have already died because of Sars Kovi II. So here it is here. This is what it looks like here. It has the membrane around the outside here. The spikes and the viral spike proteins embedded in it. And on the inside, here is the capsid here. This helical structure here is the spiral capsid, also protein. And inside there is the genome, which is an RNA strand, which is a positive RNA strand. That is, when it enters the cell, the host cell, it acts as messenger RNA so that it starts to get translated by the host cell. So this is the way I showed you before, but here is the way in which it enters the cell. It enters through a vesicle, which swallows it effectively from the outside. And what it does, it induces the formation of clathrin proteins, which buckle and then form a vesicle. So this is entry by endocytosis. And I will just mention to you one of the reasons that ivermectin and also hydroxychloroquine, these sort of things, became treatments of interest. They don't work, but treatments of interest is because they are known to affect these processes of endocytosis, but they don't affect the entry of Sars Kovi Ii, unfortunately. So effectively, the whole virus is swallowed like this and makes its entry into the cell. In the cell, the virus is ejected from its envelope and then from its capsid as well inside the cytoplasm. And this is the messenger RNA genome here. And the messenger RNA genome does two things. Immediately, the messenger RNA is translated by ribosomes, and the first thing it forms is its own replicase, its own RNA polymerase. It's a viral RNA polymerase that immediately begins to produce many, many copies of the viral genome. Then, in addition to that, the messenger RNA is translated to produce all of the viral proteins. And these are targeted so that they embed themselves first of all, in the endoplasmic reticulum, and then in the Golgi apparatus. Now go back and remember, this is how things are processed and trafficked inside the cell. And the endoplasmic reticulum and the Golgi apparatus are induced to produce what are effectively small vesicles. And these are small vesicles here. This is membrane from the Golgi here. And it's embedded in it are proteins which have been produced and matured in the endoplasmic reticulum. And the Golgi packages the viral genome together with that host membrane with its viral proteins embedded in it, and then exports that as a vesicle. So it exports it to the cell membrane, and the viral particles is released. The cell itself can actually survive for quite a long time until its own functions are so compromised that the cell dies. But this system here allows it to form a viral factory for a long time. It will continue producing viral particles. Okay, so I think that this basically just explains what I've just said. Okay, so having said that, let me just have just a quick word about the vaccine structures, the vaccine types. I want to point out, I just want to go back here, first of all. All of our vaccines operate by targeting these spike proteins here in one way or another. One set of the vaccines are all what are called old-fashioned vaccines. What they've done is they have taken viral protein like this, and they have modified it in one form or another to effectively teach the immune system what these proteins look like. So if you get a Johnson & Johnson vaccine, what you're effectively getting is highly modified viral protein, which your immune system then recognizes as being foreign and produces antibody to. And in the process, your immune system forms memory of what that protein looks like. If ever you're exposed to the virus, then hopefully your immune system produces antibody to wipe the virus out. So that's kind of Johnson & Johnson vaccine. But the other vaccines, which are the Pfizer and the Moderna vaccine, are entirely new kinds of vaccines. They have been in development for many years for this specific reason. And that is that all of the research has gone into developing a system of delivery. So this is where most of the research went. It was in developing a membrane structure, which is like an artificial membrane, artificial vesicle, if you like, which is able to recognize the cell membrane of the person that it's injected into, unite with it, and inject its contents. So all of the research into these modern vaccines, the Pfizer and Moderna, actually went to producing this carrier structure, which would unite with the cell and inject something into the cell. And what they are injecting is messenger RNA. And it's a short piece of messenger RNA. And all that it codes for is this spike protein. So what they're doing is turning a few of your cells. It's mostly in the region where you get the injection, and a small amount in the lymph nodes around the injection site. The carrier vesicle, which of the vaccine, has in it messenger RNA, and it's injecting that messenger RNA. But the messenger RNA is not for the virus. It's just for the protein. It's just for this protein. And that protein is then expressed by the cell. And when it enters into circulation in your blood system, your immune system sees that spike protein floating around on its own. It sees that spike protein as being foreign, and it will produce antibody to that spike protein, and it will form memory of what that structure is, so that if ever you're exposed to the COVID vaccine, you produce a reaction which eliminates it. So this is the important thing about these mRNA vaccines. It's very, very easy to produce this messenger RNA, this short little piece of messenger RNA. You know now how messenger RNA works. If another virus comes along, we've got the carrier system already. All we need to do is put in this little piece of messenger RNA, which codes for some aspect of the new virus, whatever it is, and we're good to go again. That's the huge advantage of these messenger RNA systems, and that is why they're being developed. And fortunately, this is one of the reasons why we could produce these vaccines so quickly, because all we had to do was find out how to make this little piece of messenger RNA. Okay, so I'm going to leave that there, and we'll finish this discussion at our next class. I wish you all a very happy Thanksgiving, and please stay safe, wear your masks. If you haven't been vaccinated, please get vaccinated before you gather together with whole crowds of other people, okay? And I will see you after Thanksgiving.